Hello everyone. We are ready with our new topic, AIDS, that is acquired immunodeficiency syndrome. In this video, we are going to introduce the disease, the causative agent, its structural details. We will also discuss about the infection cycle of AIDS along with the cure. Although there is no possible cure of this disease, yet we have some antiretroviral therapies that we have discussed in this presentation. So we all know that AIDS is caused by a virus, that is human immunodeficiency virus or HIV, which mainly attacks the immune system. And in immune system, especially the cells which get affected by HIV, they are the T helper cells, which gives you the protection. Although the disease is chronic, so the progressive destruction of the immune cells takes place, which makes your body susceptible to the infections, normal infections also. So the immune system, it gradually, uh, with time, it get weakens, and the simple infections may be the cause of the death. Now, we all know that AIDS, it represents the advanced stage of HIV infection. In the beginning of HIV infection, it is difficult to reveal the symptoms. So when the HIV infection it comes to the later stages, then that stage we call it as AIDS. And for the very first time, AIDS was recognized in the year 1981, and it is a global health problem, which is recognized as a global health problem by WHO. This virus, as far as we discuss, its family is Retroviridae, and the genus is Lentivirus, which causes slow but chronic infection which persists for a longer period of time. Now, mainly there are two types of viruses: HIV virus, HIV-1 and HIV-2. Both virus, they can infect the immune cell, that is CD4 immune cell, that we also call as Th cell, and causes their destruction. However, out of these two, HIV-1 and HIV-2, HIV-1 is most widespread worldwide and is highly infectious. It progresses faster if left untreated. Now, HIV-1 is also responsible for global HIV pandemic, while HIV-2 is less common and mainly found in West Africa. See, it has a lower transmission rate, means it do not spread with the efficiency as HIV-1, and the progression of the disease is also very slow. So the symptoms are almost negligible, so we call it as asymptomatic for a longer period of time. So the faster symptoms appear in case of HIV-1 as compared to HIV-2. Now, what causes the spread of HIV? So an HIV infected person, if it get in contact with with a normal individual during unprotected sex, or the infected blood transfusion reaction takes place, it can also spread from mother to child during pregnancy, during delivery, and during breastfeeding, because all these includes the exchange of body fluids. It also may be transmitted by sharing the needles of the HIV infected person. So the HIV infection may be spread from one individual to other through all these means. If I talk about the structural details of HIV, it is a complex enveloped spherical virus. Enveloped stands for that it has some extra covering around it. So it consists of an outer lipid envelope, which is obtained from the host cell membrane. And in this envelope, there are glycoprotein spikes, GP 120 and GP41. So GP120 and GP41, as you can see here in the figure, it is clearly mentioned. The GP140 and GP41, GP120 and GP41, these two glycoproteins, they are important to establish the infection cycle. Now GP120, if I talk about, it binds to the CD4 receptor of the immune cells. The immune cells, CD4 plus cells, the CD4 receptor binds to GP120. However, GP41, it allows the fusion with the host cell membrane. So we will discuss this infection cycle later as well, but the glycoproteins have very important role in establishing the infection by HIV. Now we are continuing the structural details of HIV in this slide also. So these glycoproteins GP120 and GP41, they help in the attachment and fusion of the virus with the host cell. Now beneath the envelope you can see that there is a green colored matrix protein P17 that provides it a structural stability, and just inside it there is a viral capsid denoted by yellow color line, and it is P24, and this P24 is cone shaped, and inside it you can see the helical structures, which is the genetic material, the RNA of this HIV. And this genome has two identical single stranded RNA molecule represented by blue color. Now in addition to this, inside the capsid you can see that there are the essential viral enzymes, three important enzymes which help the virus to establish the infection are reverse transcriptase. This reverse transcriptase help in conversion of the RNA, which is the genetic material of HIV, to DNA, which is required to establish the infection cycle in the host, which is the human being. Integrase help in insertion of the viral DNA into the host genome, while protease is required for the cleavage of the polyproteins during the assembly process of the virus. Now here you can see you have the figure which actually shows you the infection cycle, and in this infection cycle you can see that we do have figure 1 and figure B, figure A and figure B. In figure A you can see number 1 is denoted, where number 1 you can see that this is the HIV virus which is coming near to a CD4 plus cell. So you can see that on the surface of the host cell there are CD4 receptors. So HIV GP120, it binds to CD4 on the target cell, and the GP41, it mediates the fusion of the HIV with the target cell. Now nucleocapsid containing viral genome and enzymes, it has entered into the cell in the third step, while in the fourth step the viral genome and enzymes are released following the removal of the core protein. In the fifth step you can see that viral reverse transcriptase, it catalyzes the reverse transcription of the single stranded RNA genome of the virus, forming the DNA RNA hybrid. And in the sixth step, the original RNA template is partially degraded, and we get the double stranded DNA. Now the double stranded DNA of HIV virus has is then translocated to the nucleus and is integrated into the host chromosomal DNA by integrase enzyme. So you can see that it is inserted into the host CD4 cells as provirus. So this is a latent phase. It is the latent infection that it stay over there as a dormant virus. So the virus, it has entered into the host, and there it will stay as a provirus. So a copy of the viral RNA has integrated into the host genome, right? Now, in case this provirus may get activated to the active virus. Now, what happened in that series of reaction when there is an activation of provirus? So, this stage where there is a formation of provirus, it is not called AIDS. When the provirus get activated and it start forming copies of itself, then we call this stage as AIDS. So, there are transcription factors which stimulate the transcription of the proviral DNA into the genomic single-stranded RNA, and after processing, there are several mRNAs which are formed, as you can see here in B figure, step number 1. The viral DNA is then exported to the cytoplasm, right? Now the mRNA has come to the cytoplasm, and there is the host cell ribosome in the cytoplasm that catalyzes the synthesis of viral precursor proteins. Now, viral proteases, the third important enzyme, it will cleave the precursors into viral proteins, and now HIV, single-stranded RNA and proteins, they assemble beneath the host cell membrane. And in the fourth step and in the fifth step, you can see that the membrane bud out, forming the viral envelope, and the viral particles are released as the after completing the maturation. The incorporated precursor proteins are cleaved by viral protease present in the viral particles. So this is how it is going to complete its infection cycle. Here are the steps given for each of the infection stages. So first, the virus is going to attach itself to the CD4 receptor. In the second one, means in the attachment step, we can say that GP 120 and CD4 receptor, they both of these are going to attach with each other. In the fusion step, we can say that GP41 is going to mediate the fusion. In reverse transcription, we can say that viral RNA is transcribed it is transcribed to DNA by enzyme reverse transcriptase. In integration step, the viral DNA was inserted into the host chromosome with the help of enzyme integrase. Latency phase is the one where the virus lies in the as a dormant molecule, as a provirus into the T cell, as we saw that it was lying over there as a provirus. Now in the activation and the replication phase, the host cell get activated and the viral transcription and new virions bud from the membrane, and in immune destruction, HIV gradually destroy the neighboring CD4 plus cells. The other cells of the immune system also get destroyed, like macrophages, dendritic cells are there. So this is how it is going to cause the infection. Here also in this diagram, it explains you all the steps in a better way. So how the attachment is going to take place, the fusion and the entry of the HIV into the cell, then reverse transcription is taking place, then integration of the viral DNA into the host genome to form the provirus, then replication is there, activation and replication is there, and finally there is assembly and release of the progeny viruses to the neighboring cells. Now, how are we going to screen that a person has been affected by HIV? So first thing is serology, where we are going to find out that whether the antibodies are present in the blood sample of the patient. In the second one, we can go for immunoassay, where HIV antigens are tested for the same. HIV antibodies can also be checked in the other body secretions like the saliva. However, a confirmatory PCR test is important to make sure that whether the infection has been established or not. Now, if I talk about the treatment, as we all know that HIV is not a curable disease, it's not curable. Still, we have antiretroviral therapies that we call as ART, and these antiretroviral therapies, they are helpful for they support the survival of the individual. So there are various steps in the infection cycle which can be targeted in order to achieve the spread, to in order to achieve the inhibition of the spread of the virus. So first are the protease inhibitors. So the if I talk about the protease inhibitor, so these they are going to inhibit these protease inhibitors are going to inhibit the cleavage of the viral polyproteins during the assembly, so that the mature viral particles are not going to spread anymore. And the example of such antiviral drug is ritonavir. Secondly, there are integrase inhibitors which do not let the uh, double-stranded DNA of the HIV, which has been formed inside the host, to get integrated into the host genome as provirus. So that integration is inhibited by integrase inhibitor. So if that enzyme is inhibited at that stage, so it is not going to cause the formation of the provirus. So the example of such drug is bictegravir. The third are the fusion inhibitors, which inhibit the fusion of GP41 with the host cell membrane, and enfuvirtide is the example. Reverse transcriptase inhibitors or RT inhibitors do not let the formation of DNA from the RNA of the HIV, and delaviridine is the example of such antiretroviral drug. So with this we have covered almost um, every part of the HIV and AIDS. Thank you so much for listening it patiently. Please remember to like, share and subscribe the videos. Thank you so much.